Welcome to today's Explainer. We are jumping right into one of the absolute most enchanting things you'll ever see in the natural world, glowing forests. For centuries, people wandering into dark woods have been totally captivated by this eerie greenish light just sort of oozing out of decaying wood. Well, today, we are diving deep into the lab research that is actively decoding how and why these fungi produce this light. We're going to look at their bizarre diets, the specific triggers, and some absolutely incredible stress responses that actually make these organisms shine their brightest. But first, I've got a really weird question for you. What do you think happens when you take a scalpel and literally attack a glowing fungus? I mean, it sounds totally counterintuitive, right? You'd naturally think that injuring a living organism would weaken it or, dim its lights. But as we're going to see a bit later in this explainer, the triggers that actually force these fungi to shine their absolute brightest are incredibly strange and totally unexpected. So stick with me, because the answer is going to completely change how you view these quiet little forest dwellers. Okay, let's get our bearings here. You might have actually heard of an old folklore term, foxfire. It sounds like something straight out of a fantasy novel, but foxfire is just the historical, everyday name for fungal bioluminescence. We're talking about the visible greenish light emitted by the mycelium, which is basically the root-like network of threads of certain higher fungi that live on rotting wood. So this isn't magic, it is pure, hardcore biochemistry. And to really understand the mechanics of it, scientists have taken this ancient folklore straight out of the dark forest and brought it right into the brightly lit laboratory. To figure out the actual roles of foxfire, researchers zeroed in on 3 star specimens from completely different corners of the globe. First up, we've got Neonothopanis namabi, which comes from the hot, humid subtropical forests of southern Vietnam. Next is an Armillaria species from the really harsh continental climate of Krasnoyarsk, Russia. And finally, Pinella scepticus, found near Ottawa, Canada, a place with a freezing continental climate. The really incredible thing here is that despite evolving in completely wildly different environments, all 3 of these species share the exact same remarkable ability to emit visible light. And the visual contrast is just striking. In standard daylight, these fungal colonies just look like unassuming white fuzz. I mean, you literally wouldn't look twice at them if you passed them on a hike. But flip the switch to darkness, and they undergo this wild transformation. They radiate this vibrant, eerie green glow. It's a continuous light, too. And sure, it might be kind of dim compared to a household light bulb, but for a tiny biological organism, it is a stunning display of raw energy. Now, if you want to study this glow, you obviously have to keep the fungi alive and happy in the lab. So scientists basically have to act like specialized dietitians trying to find the ultimate menu. In the research we're looking at, they tested out different organic nutrient bases, things like potato broth, malt extract, sucrose, and yeast. They cultivated the fungi on solid agar plates just to establish a solid baseline. They really wanted to answer one fundamental question. Does the food that makes a fungus grow the fastest also make it glow the brightest? Just look at the sheer speed of our tropical specimen. When they tracked physical growth, specifically the colony diameter over 25 days, there is a very clear outlier. The Vietnamese fungus Neonothopanis nambi is the undisputed champion of rapid growth. It just shoots up, completely taking over the space in just a few days, totally leaving the slow and steady Canadian and Russian fungi in the dust. Naturally, you'd think all that massive growth would equal a massive amount of light, right?
Well, science actually loves a good paradox. Turns out fast physical expansion absolutely does not equal a bright glow. It was the exact opposite. Neonothopanis nambi, our super fast-growing champion, was incredibly dim on almost all the diets they tested. It basically dumped all its energy into just spreading out as fast as possible. On the flip side, the slower-growing Pinella scepticus from Canada really took its time expanding, but it was the glowing champion, radiating this intense light for over 40 days on most of its food. So the diet really dictates the balance. It's this fascinating biological trade-off between growing and glowing. The real crux of this is captured perfectly in a direct quote from the researchers. They noted, Visible glow was observed in darkness in Neonothopanus nambi mycelia when the growth of the colony slowed down or stopped. That is a profound takeaway. The fungi aren't just leaving the lights on 24/7. Fungal luminescence actually reaches its absolute peak right when physical expansion hits a brick wall. The second the fungus stops spending its energy on building new real estate, it suddenly shifts all that saved energy into producing light.
To really push the boundaries of this discovery, the researchers decided to mix things up. They moved from solid agar to liquid media, a process called submerged cultivation. They dropped the fungi into liquid nutrient broth and kept them constantly stirring. Under these new conditions, our fast-growing Nambi formed into these weird little rough spherical pellets or globules. But there was a major catch. The glow was terrible. It was extremely sluggish, registering a measly 50 to 62 relative light units per second on their instruments. They needed to find a way to wake this fungus up. And oh man, wake it up they did. Through what honestly seems like almost accidental tinkering, they hit on a trigger that resulted in a staggering increase in light emission. We are not talking about a 10% increase, not even double. We are talking about a massive 1,000-fold increase in brightness. So how exactly did they pull off that unbelievable number? They introduced something they called the water shock. The researchers basically took those dim, sluggish fungal pellets out of their nice, comfortable, nutrient-rich broth, and they just dumped them straight into plain, deionized water for 14 to 18 hours. They essentially starved them. Actually, scratch that. They completely shocked their environment. And the result? An absolutely explosive luminescent reaction. Now, the physical pellets themselves don't look any different, but the light emission is night and day. Before the wash, the sample is practically pitch black, but that exact same sample, right after being shocked with the deionized water, it is radiating intensely. By suddenly pulling the rug out from under its environment and causing massive osmotic stress, the researchers essentially hacked the fungus. They forced it to crank up its bioluminescence thousands of times brighter than it ever naturally would in that liquid state. Okay, but water shock wasn't the only trigger. And this brings us right back to that bizarre question I asked at the very top of our explainer. The researchers wanted to test the ultimate stress response: mechanical damage. What happens to the glow when the organism is subjected to direct physical violence? Well, here is the dramatic step-by-step process they used. First, they took a fungal pellet that had essentially died down. It had lost its glow entirely over time and just looked totally dormant. Second, they literally went on the attack, slicing the pellet into pieces with a scalpel. And third, the glow didn't just casually reappear, it violently spiked, jumping 20-fold just a few hours after the injury. They were literally able to kindle a completely dead-looking fungus back to glowing life just by causing it extreme physical trauma. When they slashed a deep cross right into the solid colony with a scalpel, the reaction was immediate. The brightest, most intense green glow concentrates exactly along those incision lines. The fungus is aggressively pouring its light-producing energy directly into the site of the wound. It is an immediate, localized biological reaction to extreme mechanical stress. It's literally acting like a glowing alarm system. All right, let's bring all this amazing data together. First, the diet dictates a very strict balance between growing and glowing. Second, bioluminescence naturally peaks right when physical expansion stops. And finally, extreme environmental stress, whether that's a pure water shock or literally getting sliced by a scalpel blade, triggers massive spikes in brightness. On a practical level, mastering these weird environmental hacks is exactly what helps researchers sustain these delicate cultures in the lab, which means we get to keep studying the incredibly complex chemistry of foxfire. But before we wrap up, I want to leave you with a thought that takes us out of the laboratory and puts us right back into the dark woods where we started. If a fungus evolved to suddenly glow up to 20 times brighter the very moment an insect takes a bite out of it, or a falling branch crushes it, what exactly is the evolutionary purpose of that natural alarm system? Is it trying to scare the predators away? Or is it shining a spotlight to attract an even bigger predator to come and eat the insect that's attacking it? It is a brilliant reminder of the complex, completely hidden mysteries that are still waiting to be uncovered in our forests. Thanks so much for joining me on this explainer, and keep your curiosity glowing.